Read out the question number 6. Which one of the following pair is correctly matched? Okay, here names of the scientists and their discoveries have been mentioned. Just you have to match or you have to select the correct answer where you will find the scientist's name and his discovery found that. Look at the very first option. Frederick Griffith discovered the phenomenon of transformation. Of course, this is correct. You will also check the other answers. Don't come to the conclusion immediately. Option A is correct. You also check the other options. Linus Pauling. As far as Linus Pauling was concerned, he was concerned with the discovery of binding up of the oxygen molecules with the sickle cells, particularly when the sickle hemoglobin is produced. Yes, hemoglobin is produced. At that time, RBCs are going to have altered structures. At that time, whether it is going to have the capacity to carry the same amount of oxygen or not. It was explained by Linus Pauling. Then Francis Crick. You all know that Francis Crick is one of the founder scientists. He explained about the double helical structure of DNA. Okay, against the Francis Crick, the discovery is being mentioned, that is proposed one gene, one polypeptide hypothesis. That's not correct. One gene, one peptide hypothesis was explained by Beadle and Tatum. It is also called as Beadle and Tatum hypothesis. It is not matching with the Francis Crick. Even you also look at the option D. George Beadle proposed the concept of inborn errors. No, George Beadle is not concerned with this. Beadle and Tatum explained about one gene, one polypeptide hypothesis. Okay, that's why the correct option A is correct. Frederick Griffith discovered the phenomenon of transformation. He conducted the experiments on mice. He conducted the experiments on mice and finally he came with the conclusion that transformation is possible when the foreign DNA is going to enter there into the organism. Whenever there is a transfer of DNA takes place from other sources, always there is a possibility of changing the characters. He conducted the experiment, series of experiments with the mice. Yes, finally he came with the conclusion. Okay. For that transformation experiment, he made use of the bacteria called Streptomyces pneumoniae. Even they are also called as Diplococcus pneumoniae. There are two strains present, R strain and S strain. By using these two strains, he conducted the experiment and he finally explained, yes, it is because of what you call as the transformation change in the character may occur. Okay. Next question number 7. Match the following and mark the correct options. Okay. There are two columns given, column 1 and column 2. Okay. You have to match the contents of the column 1 and the column 2. Okay. Now, column 1, it goes like this: oncogenes, single circular DNA, splicing, Jacoban monad. Okay, in the column B, the explanation is given like this. Number 1, causes AIDS. Number 2, Lacapron concept. Number 3, component of bacterial chromosome. Number 4, processing of mRNA molecule. Number 5, when activated, it causes the cancer. Look at this. They are particularly oncogenes. Oncogenes are the genes that are present in suppressed form. They are present in every human being. As and when these oncogenes are getting activated, definitely they leads to the cancer. Okay. Option 1 matches with what you call as number 5. Okay. Then single circular DNA. Okay. Usually DNA molecules, they are usually present there in what you call as bacteria, particularly prokaryotes. In prokaryotes, there is a double-stranded circular DNA molecule is present. And you look into the bacterial cell, say for example, there is a single double-stranded circular DNA molecule is present. Sometimes, in addition to this, you will also have extra DNA molecules. If they are present, we refer them as extra chromosomal DNA molecules. They are referred as plasmids. But plasmids are not compulsorily present in the bacteria. Remember, only some bacterial species may have the plasmids. But usually, most of the bacterial species cells are having single double-stranded circular DNA molecules. Okay, here it matches to single circular DNA molecule. It matches with component of bacterial chromosome. Means number 2 matches with what you call as number 3. Then splicing. Splicing. RNA splicing, what we call it. You must remember that there in what you call as DNA of eukaryotes. DNA of eukaryotes usually contains 2 segments. Some of the DNA segments are usually called as exons, and some other DNA segments are called as introns. Exons and introns, they are present one after the other. Means there are so many exons present. This may be exon 1, this may be exon 2, this may be exon 3. Okay, like this. Even there are also so many intron sequences. This may be intron 1, this may be intron 2, and so on. The difference between the exons and introns is usually exons are the regions. Remember, these regions will code for the protein synthesis. They are going to be coded for the protein synthesis. Means here there are certain genes present, and these genes are going to code for the protein synthesis. Even in the exon 2 also there are genes present. They are again used for the protein synthesis. Like that, even exon 3 also contains the genes used for the protein synthesis. What about the introns? Introns are the places where DNA segments that are present are not going to be used for the protein synthesis. Introns will not be used for the protein synthesis. Okay. Usually what happens is, at the time of transcription, particularly in eukaryotes, as and when the transcription occurs, RNA polymerase is going to synthesize RNA on this entire DNA molecule. It synthesizes RNA both on exons as well as introns. Assume that these are supposed to be the intronic regions of the RNA. These are the exons of RNA. This may be exon 1, this may be exon 2, this may be exon 3. Okay, these are supposed to be the intron regions. Intron 1, intron 2, like this. Okay, this RNA that is going to be present having both exons and introns is usually called as heteronuclear RNA, HNRNA. Eukaryotes, first of all, HNRNA takes place. It is going to be synthesized. Later, what happens is splicing up of the RNA takes place. In the process of splicing, intron regions are going to be removed. Assume this is one intron region. Yes, it is going to be removed. This is the second intron region. This is also going to be removed. And only exon regions are separated. This is one exon. Okay, you may consider it as exon 1. And this is supposed to be the second region of exon present on mRNA. And this is the third region of exon that is present on mRNA. Okay, these are the exon regions of the RNA. They are going to be attached to one another, ligate. This is all going to happen in the process of what you call as RNA splicing. We call this process as splicing. Okay, splicing usually leads to the formation of messenger RNA. Here, HNRNA, heteronuclear RNA, is transformed into messenger RNA through the process of splicing. You may ask the question directly that why not in eukaryotes directly messenger RNA is going to be produced? The reason is, yes, this messenger RNA, once it is produced, it is having a very shorter span of life. Okay, it is not going to survive for a very long period. As soon as it is produced, yes, it has to undergo the process of protein synthesis. It has to start synthesizing its proteins. It is not going to be active for a very long time. Its lifespan is very less. That is why only when a proper protein is going to be synthesized and needed, at that time only the splicing occurs and the mRNA is going to be produced from the hnRNA. That is why we always call hnRNA as a precursor molecule of mRNA. Precursor molecule means it is the molecule that is going to be synthesized prior to the synthesis of mRNA. Remember, in eukaryotes, mRNA is not directly produced on what you call as DNA. Now, we call this concept as a split gene concept. Usually, there in eukaryotes, genes are not continuously present, particularly in the intron regions. Genes are not present. Genes are just restricted only to the exons. That is why we refer this concept as split gene concept. Split genes means yes, here genes are present, here genes are present, here genes are present. Only where the exon regions are there you can see the genes. Okay, they are not continuously present there on the DNA. Okay, with this, what is the correct option here? Correct option is yes. You have to match it. You have to match. Even Jacobite monad, option 4 in the column A. Jacobite monad, you all know that Jacobite monad did some wonderful work in molecular biology. Particularly, they explained about the structure of mRNA, messenger RNA. What about the regions which are there in the mRNA? The structure of mRNA was explained for the first time by Jacobite monad. Even apart from that, Jacobite monad are also concerned with another important work, that is lactoperon. They explained lactoperon concept for the first time. When the genes express, when the genes are not going to expressed. Okay, how exactly gene expression or regulation of the gene activity takes place there in the prokaryotes. It was explained for the first time by Jacobite monad. Okay, by looking into these concepts, you have to match. Okay, your oncogenes, which are going to cause the cancer when they become active. Okay, one matches with 5. Then single circular DNA molecule, it is nothing but component of bacterial chromosome. Splicing, it leads towards splicing, it leads to processing of mRNA molecule. The Jacobite monad, okay, they are concerned with the explanation of what we call as lactoperon concept. You match them, you will find the correct answer. Okay. Next one. Number 8. Question number 8. Which one statement regarding Okazaki fragment is correct? I already told you what does exactly mean by Okazaki fragments. Okay, when the DNA replication is going to occur at that time, yes, on one of the strand, synthesis of DNA takes place continuously. Okay, here DNA replication has a direction. You all know that it starts from 5 prime. It starts from its 5 prime and it proceeds towards the 3 prime. Okay, on this particular strand, DNA synthesis takes place continuously because here DNA synthesis is taking place towards the fork. On the other strand, DNA synthesis takes place away from the fork. At that time, DNA synthesis occurs discontinuously in the form of small fragments. We call these small fragments as Okazaki fragments. Say for example, these are supposed to be the Okazaki fragments. Okay. Look at this and Find out which is the correct option. Option A, they are double helical fragments of DNA. No. Option B, these are single stranded fragments of DNA. Okay, of course it is correct. Even you also check up the other options. Option C, they are the fragments of mRNA. No, not correct. Then D, these are the fragments of tRNA. That is also not correct. Here option B is correct. They are supposed to be the fragments of single stranded fragments of DNAs. Option B is correct. Question number 9. Exon part of mRNA has a code for. I just made the mention that just now I told you exons are nothing but what? Exons are supposed to be the regions which are coding for the protein synthesis because exon regions has got the genes. Okay. Usually on the exon regions only RNAs are going to be produced, and on these mRNAs there is a message written for the synthesis of protein present. That is why we always say exons are usually meant for what you call as the synthesis of protein. When these mRNAs are going to associate with the ribosome, their coded information is present in the form of triplet codons. Ribosome reads these triplet codons and it is going to assemble the amino acids so that a proper protein is going to be synthesized. Always remember, exon part of mRNA as a information or code for protein synthesis. Option A is correct. Then, next one, question number 10. Which of the following is not correct? There are 4 options, but one of the option is not. Only one option is not correct. Only one option is not correct, but the 3 options are correct. You have to choose which one of the option is correct. Okay, the answer goes like this. Option A divided by T is equal to 1. Then B, look at the option B. It is A plus T is equal to G plus C. Then option C, A plus G is equal to C plus T. Then option D, none of this. Okay. Means if any of the above 3 answers are not correct, at that time you have to select the option D. First, you have to check whether the first 3 options given here are correct or not. Yes, first, you look at the option 1. You want to know that there in the DNA molecule, there are 2 strands present. Okay? And these 2 strands are having complementary nucleotides. Particularly the nitrogen bases which are present are complementary to one another. Suppose if adenine is present, thymine is present, cytosine is present. Again, adenine, guanine, thymine, cytosine, guanine, like this. If you know the basic sequence of one particular strand, you can always predict the basic sequence of the other strand because they are always complementary. You know that adenine pairs with thymine, thymine pairs with adenine. Yes, cytosine means cytosine always pairs with guanine. Again, adenine means thymine, cytosine means guanine, thymine means adenine, cytosine means guanine, guanine means cytosine. Okay. It's here, amount of adenine is equivalent to thymine. Like that, amount of guanine is equivalent to amount of cytosine. Suppose if there are 100 adenine molecules present, equivalent to the adenine, there are 100 thymine molecules present. If there are 350 guanine molecules present, equivalent to that 350 cytosine molecules present. The amount of the guanine and cytosine molecules are always same. Okay. Now look at the options here. Suppose if you consider that there are 100 adenine molecules, just assume there are 100 adenine molecules. Okay, equivalent to 100 adenine molecules. What about the thymine? Amount of adenine is equivalent to thymine. Yes, approximately there are 100 thymine molecules present. Okay, the ratio of A divided by T is always 1. Means this is correct. also look at the option B. A plus C is equivalent to G plus C. Consider the same. Here there are 100 adenine molecules present. Okay. Then what about the thymine? Okay, thymine is equivalent to adenine. Yes, it is also 100. This is equivalent to G plus C. What about the G plus C? Look here. If guanine is 350, then cytosine is also 350. Okay, you sum up these values. 100 plus 100 means 200. 350 plus 350 means it is 700. Okay, this is not correct. A plus T is not equivalent to G plus C. Also look at the option C. Look at the option C. A plus G is equal to C plus T. Okay, assume that adenine is 100. Guanine is 350. Okay. Then equivalent to that, yes, C. When guanine is 350, cytosine is also 350. When adenine is 100, thymine is also 100. Okay, it becomes 450 on LHS. On RHS also, it becomes 450. So that 450 is equal to 450 means, okay, the ratio is always equal. This one. Yes, you can say that option C is also correct. Then none of these. Yes, what is the wrong answer? We know option B is not correct with reference to this particular question. That is why option B is supposed to be not correct with this particular question. Okay. In the next class I will continue, and I am just going to explain some basic concepts in the initial classes. Okay, as and when the class proceeds, you try to understand some of the points, and at that time I am going to fast and I am going to tell you some concepts, and as and when you find difficulty, you can contact me over the phone. Okay, no problem. Thank you.